Alright, good day everyone. Welcome back to our YouTube channel. Today on this channel we're going to be talking about gel electrophoresis. And in gel electrophoresis we're going to be illustratively dealing with the two main types of gel electrophoresis. Don't forget to like and subscribe to our YouTube channel. Electrophoresis can be divided into electro, meaning electricity, and phoresis, meaning to carry. It's the movement of molecules like DNA, RNA, and protein mobilized by an electric field. In any gel electrophoresis setup, there are two electrodes, a positive electrode near one side of the gel and a negative electrode on the other end. When the power supply comes on, these electrodes create a difference in charge on the two sides of the gel. This creates an electric field. How electrophoresis separates molecules is determined by the molecule's size and charge. Charge causes the molecule to move in the gel as it is attracted to the opposite charged pole. Positively charged molecules move towards a negative electrode, while negative charged molecules are pulled towards a positive electrode, while the neutron molecules do not move at all. Therefore, DNA molecules being negatively charged will move towards the positive electrode.
But scientists want to separate DNA fragments not based on charge but by size. So they put them into a gel full of irregular holes like a sponge. Therefore, moving through such complex holes, the size of the fragment determines how fast they move. Short or small pieces will move faster, while larger pieces will move slower as they encounter hurdles due to their size. The small pieces will move faster to the opposite side of the gel, while the large pieces will move slowly. Therefore, using this, Agarose gel electrophoresis can be used to separate molecules.
In creating agarose gel, we dissolve agarose in an electrophoresis buffer, boil it, and then turn it into a casting tray. Then we place a comb at one end of the gel. Once the gel cools and hardens, we pull it out, leaving the little holes called wells in the gel. We then transfer the cooled gel to an electrophoresis chamber. We cover the gel with running buffer, helping it to conduct electricity.
Then we pipette our samples into the wells. We'll cover the lid up and turn on the power. We call the region in which these samples travel from the well to the far side region a lane. While these protein molecules moving in a lane consist of similar packed molecules of same size and speed, called a band, often in a straight line. How quickly a DNA sample travels in comparison to another let us know how big they are compared to one another. For example, the protein molecules in band A have moved faster than the protein molecules in band B. Therefore, the molecules in A are smaller than that in band B. Agarose gel electrophoresis helps us in molecular biology, allowing us to visualize and understand protein molecules as well as DNA and RNA.
SDS-PAGE, known as sodium dodecyl sulfate polyacrylamide gel electrophoresis.
In this form of electrophoresis, proteins are separated in terms of their molecular weight in a polyacrylamide solution. For sample preparation, a loading buffer containing SDS and beta-mercaptoethanol blue is added to the protein samples. Protein samples can be biologically derived from various means like viruses, tissues, or purified proteins. When the samples have been added, the samples are heated at 95 degrees Celsius for 5 minutes.
Proteins are long chains containing amino acid residues. Amino acids are organic compounds containing amine and carboxyl functional groups with a side chain. They are formed by linking amino acid with peptide bonds. SDS denatures proteins by disturbing the hydrogen bonds and its interactions.
For gel preparation, a separating gel solution with pH of 8.8 and a stacking gel solution with pH of 6.8 are used. They contain acrylamide, bisacrylamide, ammonium prescopate, tamed, Separating gel is poured first with ammonium prescopate, initiating the free radical with 10. Biacrylamide joins these chains together, serving as a linker. The pore size of the gel is related to the ratio of acrylamide to bisacrylamide.
The stacking gel is then poured on top, and also the comb is put into the top of the gel, leading to the formation of wells on the gel. Once the gel completely polymerizes, the comb is removed and the gel tray is put directly between the two electrodes. The gel is placed into the electrophoresis system, pouring in a running buffer of pH 8.3. It allows the conduction of electricity throughout the system.
The samples are pipetted into their respective wells. After this, an electronic field is applied, causing the migration of protein molecules from the negative electrode to the positive electrode. Smaller protein migrates quickly through the mesh of the gel, while larger proteins are retained in the mesh of the gel.
At the end of the electrophoresis separation, all proteins are sorted by size and can then be analyzed by other methods and techniques. After removing the glass plate, the stacking gel is discarded and the separating gel is stained. Proteins are detected as blue bands in the background. Upon this staining, the protein bands can be seen within 10 to 20 minutes.